A very good evening to my class 12th, who is all of those who are going to appear for board exam this year. So, I really am making this video after such a long time, but yes, now it is high time to start revision. And although I have explained this chapter in a very complete detail, every topic, so I will put the link in the description box. You can go for detail to those videos. Here I am taking specifically a quick revision part. I have taken all the important lines topic wise from the NCRT, and we are going to stick with that, and we are going to solve our in-text question also one by one. So daily I will try to put one video for you so that you can revise this chapter, and this chapter should be crystal clear to you, because this is now principal basis and molecular, both are very important chapter for us this year, right? So let's quickly start with our chapter, that is molecular basis of inheritance. As you know that now though you have given pre-boards also, right? So there is a clear picture, one thing in your mind, or it should be in your mind, that there are in this chapter we have talked about two molecules, that is one is DNA and another is RNA, right? And we have talked about the genetic material. So here what is so important for us, first is what is DNA? It is deoxyribonucleic acid. Now what is actually DNA made up of? We know that DNA is a long molecule, right? So it is not made up of, it is a polymer. So it is made, it is a molecule which is made up of a polymer of nucleotides. And कौन से न्यूक्लियोटाइड्स? डीऑक्सीराइबोन्यूक्लियोटाइड्स. बच्चा deoxyribose, it is a sugar, right? And when we attach sugar with your nitrogen base, plus we add one phosphate, then it completes our nucleotide. So we know this is our basic, that nucleotide, one nucleotide, it is made up of what? One nucleotide, it is made up of sugar, it is made up of nitrogen base, right, which can be purine and pyrimidine, plus it must have your phosphate, that makes your one nucleotide. But here we are saying polymer means many nucleotides. कौन से nucleotides? Sugar is deoxy, so deoxyribonucleotide. So DNA क्या है? It is a long polymer, it is made up of a chain of, है ना, रट्टा तो नहीं लगा सकते, cram we can't do cramming part, so we have to understand it is a polymer, it is not a monomer, it is made up of long chain of nucleotides. सो लेंथ ऑफ डीएनए, ये अगर मेरा डीएनए है, हाउ मैनी बेस पेयर्स दे आर अटैच्ड ऑन दिस डीएनए. अब ये डीएनए बनाया तो मैंने बहुत इजी फॉर्म में है, बट एक्चुअली these all are my nucleotides. One nucleotide, second nucleotide, third nucleotide, they are making this structure. So number of nucleotides, or basically base pair, they actually give us length of DNA. And number of base pairs, number of base pairs, इसको तो mark कर लो. You should mark it in your NCRT right now. I have taken all these points from your NCRT only. The number of base pair, very important. हम बोलते हैं क्यों लर्न करना है इनको, this is of no use, but you are wrong, because at the end you are going to understand that why these base pairs, they are so important for us, because this is the characteristic. Every number of base pair, it is the characteristic of every species. हर species का बच्चे एक अपना नंबर है. जैसे हमारा ह्यूमन बींग्स का नंबर है एक बेस पेयर, हमारा जो डीएनए है वो कितने बेस पेयर से बना है. हेप्लॉइड नंबर की बात करें तो 3.3 × 10⁻⁹. अगर डिप्लॉयड हो जाए, डिप्लॉयड that will be 6.6 into 10 raised to power 9 base pair होगा. I am going to ask you this question at the end. E. coli में कितना होता है? 4.6 into 10 raised to power 6. Lambda phage में 48502, लिटिल बिट थोड़ा लेस होता है हमसे. एंड 5174 फेज में वायरस में दैट इज़ योर 5386. 5386 इज इन लैम्डा 174. सो ये तो आपकी टिप्स पे होने चाहिए, राइट. ह्यूमन बींग्स में देयर इज 3.3 हेप्लॉयड नंबर है बच्चों, 6.6 हमारा डिप्लॉयड है. इकोलाई में 4.6 है, लैम्डा फेज में 48502 एंड 5174 phage में 5386. So these are our number of base pairs. Now DNA तो बात कर लिया, definition is perfectly done. Who discovered it? First time it was not Watson and Crick. यहाँ पे one word MCQ हम गलती करके आते हैं बिकॉज दिमाग में सिर्फ वॉटसन एंड क्रिक घूम रहा होता है. बट बच्चा फर्स्ट टाइम DNA was discovered by Frederick Michener. Frederick Michener in 1869. He was the scientist who gave us that there is something acidic which is present in your nucleus. He said there is some acidic substance which is present in nucleus. That time he did not know about DNA. He gave us the term न्यूक्लियन. बच्चा न्यूक्लियस में जो प्रेजेंट है तो उसके बेसिस पे उसने न्यूक्लियन नेम दे दिया. सो DNA was who discovered DNA? Frederick Michener. And which kind of substance? अब बहुत सारे बच्चों को when I ask that DNA is basic, DNA is acidic, it is an acidic molecule. That is the reason जब हम packaging की बात करते हैं, it is packaged by positive charged protein, basic protein. So itself it is a acidic substance. Now there is a this diagram given in your book, and इस डायग्राम से पता नहीं क्या फोबिया है, एवरी वन ऑफ यू हैव. इट इज़ वेरी इजी टू मेक. बच्चा क्या बनाना है न्यूक्लियोटाइड में? शुगर, राइट, नाइट्रोजन बेस, एंड देन यू हैव टू पुट फॉस्फेट विद CH2P, राइट. तो पूरा भी लिख सकते हैं. सो फर्स्ट मैं बनाती हूँ पहले आपके सामने. देखो, वी विल मेक आवर शुगर. डन. नंबर दिस 1 2 3 4 एंड दिस कार्बन इज 5 हमेशा रिंग से बाहर होता है. नाउ विच नाइट्रोजन बेस इज अटैच्ड? फर्स्ट नाइट्रोजन बेस इज ऑलवेज अटैच्ड ऑन फर्स्ट कार्बन. सो यहाँ पे एडेनिन, एंड on fifth carbon we have CH2, and with CH2 we have phosphodiester bond, that is your phosphate. Now with this there is another nucleotide. It is having which base? Thymine. Now there is your 5 carbon, right, and this 5 carbon will make this phosphate, it is going to make a bond with your OH, right. So here comes your OH. It will make a bond with the OH of third carbon. Done, right. Now same here. Here is phosphate making bond with your hydroxyl group of third carbon. But at the end, at the last, which carbon is free here? That is fifth carbon. Fifth phosphate is free. इसके साथ कोई bond नहीं बन रहा. Here which carbon number is free? Third carbon, which is having OH group. That is the reason we are giving polarity. क्या direction? Polarity means in which direction this chain or this strand of DNA will run. This strand of DNA will run in which direction? What is the direction? Here 5 end is free, and the third dash OH is free. Here 5 dash phosphate is free. So this is 1 2 3. You can make a chain of 4 to 5 nucleotide. So this chain is known as polynucleotide chain. So बच्चा इस chain से घबराना नहीं है. First thing you always have to do is number your ring. 1, 2, 3, 4 and fifth carbon. Fifth carbon is having CH2 and phosphate. Now here they are not forming any bond, so fifth is free. Here our 3 dash is free. Clear? Next is Watson and Francis Crick. Meshner discovered in 1869, and Francis and Watson, they gave us proposed a double helical model of. So who gave us the double helical model? Ki DNA is arranged in your double helical, 2 strands it has. That was proposed by Francis Crick and Watson. They use the data. ये लाइन हम मिस कर देते हैं एनसीईआरटी में. ओपन योर एनसीईआरटी एंड मार्क दिस लाइन. They obtain the data, they use the data which was obtained from X-ray diffraction, and this data was produced by Morris, Wilkins and Franklin. दिस इज योर नीट क्वेश्चन ऑल्सो बच्चा. एक्सरे डिफ्रैक्शन का जो डाटा था वो वॉटसन एंड क्रिक ने नहीं फॉर्म किया था. इट वॉज प्रोड्यूस्ड बाय मॉरिस विल्किंस and Franklin. On the basis of that data, the Watson and Crick, they proposed double helical model of DNA. What was the so important? ये 3 marks में question आता है. Write down the characteristics of double helical model of DNA. The most important feature of the model was complementary base pairing, and base pairing rule was given by Erwin Chargaff. He observed that in double standard DNA, double standard matlab this is one strand, there is second strand. Now how are the two attached? We are saying they are present in the helical form. So how do they are attached? So they said, first thing, the ratio between adenine and thymine and between guanine and cytosine, that will be always constant. Right? Means the number of, इसका मतलब क्या है? The number of adenine, the ratio है ना, the number of adenine will be always equal to number of thymine, or you can say A by G will be all A by T will be always equal to your 1, and G by C will also equal to 1. Their ratio will be always constant. You cannot change. जितना इस स्ट्रैंड में एडेनिन होंगे उतने ही इस स्ट्रैंड में थायमिन होंगे. जितने इस स्ट्रैंड में गुआनिन होंगे there will be cytosine. So their ratio will be constant. So they said adenine ratio, adenine by T always equal to 1, G by C always equal to 1. Why this is so? Because adenine base pair with thymine and guanine base pair with 3 hydrogen bond with the cytosine. Now how to draw it in this chain? I already told you that this is our deoxyribose sugar. This is which carbon? Third carbon. This is first carbon with the nitrogen base. There is fifth carbon with the phosphate. Now here fifth carbon is free. Here 3 carbon is free. So if one strand is running in 5 dash to 3 dash, other will run from 3 dash to 5 dash. तो ये हमेशा हम रिंग ऐसे ही बनाते हैं, because that is why the first carbon faces your adenine, right? So adenine base pair with thymine, guanine base pair with cytosine. So as you can see here, fifth carbon we having phosphate, it is forming bond with your third carbon having which group? Hydroxyl group. Try it and I guarantee you can easily make this polynucleotide chain. This is already given in our NCERT, but sometimes it is very confusing for us to make that. So you can refer to this diagram and you can easily make it down. Only you need to understand that nucleotide is a sugar plus nitrogen base plus it is having phosphate with the fifth carbon. This is the concept. इस concept को change नहीं करना है. If you will stick to it, you will definitely draw this, right? So packaging, next there is important thing is about the packaging. See, in prokaryotes we always know that there is no well defined nucleus, because prokaryotes, they are very primitive organism, so we never draw nucleus there. So DNA जो है, that is organized in a very kind of large loops, that is which, what do we make in a bacteria, this kind of situation, which is known as nucleoid. So here is what your DNA is present with the positively charged proteins in prokaryotes. But it is not the easy scenario in the eukaryotes. I have discussed here very shortcut diagram, but if you want to understand the concept, then refer to my detailed video of packaging of your DNA, where I described complete about the nucleosome, and then come to this video, okay? Because here I am only and only taking the points of your NCERT, right? So that you can produce well in your exams. Now in eukaryotes, we say DNA is wrapped around positively charged what? Histone proteins, and there is these octamer. These histone proteins are actually a histone octamer structure. Very important point to refer. हमें पता ही नहीं होता है histone present कैसे है. So they are present in a octamer form. Into a octamer is we use for 8 into a structure. So this complete structure, मतलब DNA भी है, 8 histone proteins भी हैं. So this complete खिचड़ी is known as your nucleosome. समझ में आया? So basically in eukaryotes, DNA इतना lengthy है, इतना length है इसका, that we need something. We need some some kind of proper you can say ball where we can wrap it. So there we are using the balls, 8 balls we are using, and these 8 balls, they are your histone proteins, because DNA is negatively charged, so we need negative always attract positive, so that binding should be strong, and this structure is called nucleosome. So as you can see that this basic is your octamer, because it is a 3-dimensional structure, and this is the main structure of core of your histone basically. And here is the basically last one extra, that is the H1 histone protein, and as you can see we have wrapped our DNA around this. So this complete structure, DNA plus histones, that is known as, अब हमें definition की ज़रूरत ही नहीं है. We know what is a nucleosome. DNA wrapped around a positively charged histone octamer making a structure. That structure is known as nucleosome. We are done with the definition. Now you should not worry about the definition. So now the last thing I will take for today is, otherwise the video will be very long, right? So I'm taking here quickly the search for genetic material. First part of this I'm taking today, rest I will discuss tomorrow. See bacha, there was the grift, very 5 marks, 3 marks, इसी कैटेगरी में आता है यह क्वेश्चन बच्चा, इसी कैटेगरी में आते हैं इसके क्वेश्चन, और घूम फिर के जस्ट उसकी स्टेटमेंट चेंज कर देते हैं, क्वेश्चन की आंसर यही रहता है. ऑलवेज रिमेंबर, ग्रिफ्ट का जो नेम है वो ट्रांसफॉर्मेशन के साथ अटैच्ड है बच्चा. ट्रांसफॉर्मेशन मतलब कुछ तो चेंज हुआ, कुछ तो एक जगह से दूसरी जगह गया, राइट? So search for genetic material में first experiment that we performed, that is the Frederick Griffith. He gave us the transforming principle. He did a lot of experiments, and he did experiments on bacteria. What was the bacteria name? Neat question. Streptococcus pneumoniae. And what is so important about this bacteria? It was causing pneumonia. So he used 2 strains of bacteria. one is rough strain and one is smooth strain. If you will want to know that what is rough, what is smooth strain, again I will give you the link. All the complete playlist I will give in description box. So he said that जो R strain bacteria थे, they were the strain which does not cause pneumonia. They does not cause pneumonia. But what was the conclusion? What was the result? He said that R strain bacteria, they have been transformed by heat killed S strain bacteria. ये line you cannot change. You cannot change the conclusion, right? What is the experiment? Transformation experiment. And what is the conclusion? What is our aim? That we want to show that there is something which have been transferred. So he said that our strain have been changed, have been transformed by heat killed S strain bacteria. So there must be something which has transferred, and they said there is transfer of genetic material. That is why this principle is known as transformation principle. But later on, Avery, Oswald Avery, McLeodant and Carty. Avery, Leodant and Carty, they discovered that, because he did not tell us. Griffith did not tell us that what is transferring. So these three scientists, ये नीट में हम गलत करके आते हैं, because we write that it was Griffith who told us that DNA is transferring. No, bacha, no. Avery, Leodant and Carty, they discovered that DNA from heat killed S strain bacteria, it caused the transformation of R strain bacteria. He said transformation हुआ है. So it means something has transferred. But it was Avery, it was Leodant and it was Carty who said that it was DNA who has been moved, who has been transferred from heat killed S strain, and that changed the R strain bacteria. बच्चे ये result लिखना end में बहुत important है. Either its question is coming in 5 marks or 3 marks, and इसको तो tips पे अभी के अभी अपने notes में mark कर लो. You should know the point. सोचना ही नहीं है पेपर में लिखते time. Most important lengthy question. Right? So now time to solve quickly few in-text question. Okay, there was a one question that can you find out why the distance between two polynucleotide chain in the DNA always remain constant? We just said that Chargaff rule says, you can quote that in your exam, that Chargaff rule ke according, pairing of purine always pair with the pyrimidine, due to which there is a uniform नहीं याद आता पेपर में, write down the constant distance between two helix. If there is purines कौन से होते हैं? A and G. Pyrimidine T and C. So A always pair with T, G always pair with C. So basically purine will always pair with pyrimidine and they will maintain the constant distance. Okay, next question was, if length of E. coli DNA is 1.36 millimeter, calculate the number of base pair. बच्चा length हमें दी है. So distance between the base pairs, distance between two base pairs usually कितना होता है? 0.34 into 10 raised to power 9 meter. Total length of DNA is given to us. Yes, it is given to us, so we have to apply this formula. Best apply this formula. Total length of DNA कैसे count करेंगे? Distance between two consecutive base pair into total number of base pairs. अब total number of base pairs तो पता ही नहीं है, because वो तो count करना है, right? So we are taking it, assuming it n. So I am taking total number of base pair as n. Length of DNA is given to me. Distance between two consecutive base pair is 0.34 into 10 raised to power 9. इसको तो लिख लो, right? Just write it down in your notebook. And then you can calculate. So n will be 1.36 divided by 0.34 into 10 raised to power 9. So your answer will be 4 into 10 raised to power 6 base pairs. Now I'm going to page number 1. What was the answer? The base pairs of E. coli is 4.6 into 10 raised to power 6 base pairs. That is our answers, right? This is our answer. But what is the formula to calculate total length equal to distance multiplied by total number of base pairs? Last is theoretically how many nucleosomes, or you can say beads, do you imagine they are present in mammalian cell? Okay, bacha, this is not confusing at all, not at all. We should know that one nucleosome, मैंने बोला है चाहे लेंथ है डीएनए की, बेस पेयर्स आर वेरी इंपॉर्टेंट. ईच न्यूक्लियोसोम हैज 200 base pairs. So can I write it like that 200 base pairs equal to one nucleosome? What is the base pairs of our cell, mammalian cell? We know that our cell has 3.3 into 10 raised to power 9 base pairs in haploid, but we will use here diploid. So it will be 6.6 into 10 raised to power. So अगर 200 base pairs equal to one nucleosome, हमें न्यूक्लियोसोम्स काउंट करने हैं. सो 6.6 कितना हो जाएगा फिर? 1/200 इंटू 6.6. 6.6 हो गया हमारा 3.2. ये 2 कट कर दो 7. सो इट विल बी 3.3 इंटू 10 raised to power 7 nucleosomes or beads, right? So tomorrow we will search that who is our genetic material, right? So I will meet you soon. Till then, please read these all these things from your NC